This is TWIS, This Week in Science, episode number 485, recorded on Wednesday, October 15th, 2014. Half as much. Hey everyone, I'm Dr. Kiki, and today on This Week in Science, we are going to fill your head with fecal transplant pills, stem cell solidarity, and cooperative Crocs. But first...
Disclaimer, disclaimer, disclaimer. The universe is stranger than you think. Even if you woke up this morning thinking, what a sane and rational world it is you live in, even if you take the regular route to grab a morning coffee, even if your day is unusually uneventful, even if this evening everything turns out the way you expected it to, everywhere around you, all the time, just below the surface of the way things appear, strange things are happening. Not that there are actually strange things going on, seeing as they go on all the time, but atomic, subatomic, quantum things, invisible-ish, quirky, quarky things, taking vastly different paths through space-time than you are likely to encounter on your way to grab a Wednesday morning coffee. Unintuitive worlds where Newtonian physics do not apply, where the ticks and tocks of time are beat out to rhythms faster than the speed of sound. Worlds without day or night or consciousness or coffee or casual conversation, and yet, despite all this, they still have This Week in Science coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kirsten and Blair. Minions everywhere.
Good science to you too, Justin and Blair, and all the minions in the world. Everywhere. Where, oh, where are you? Everywhere. You're everywhere, science fans. All right, everyone. This is This Week in Science, and we are here to talk about science. Science. Yeah, we're going to talk about science. I have a whole bunch of stories for you this week. Stories about stem cells, new uses for nerve cells, and a competition between rigs and reefs. Yeah. Justin, what do you have?
I have fecal transplant pills exploiting females for science, an antibiotic probiotic turkey of an idea, and why this ain't the worst California drought in history.
Oh, fascinating. I'd love to hear that. And Blair, tell me a bit about yours.
Let's see, I have an awesome story about crocodiles, another about how birds are better at flying than we are, and sticky gecko feet.
Do we have to have a science study about why birds are better at flying than we are?
Not like flap our own wings.
It has to do with the wings.
A bird versus an airplane is what I meant.
Or versus a helicopter.
Yeah.
Helicopters not meant to fly.
Yeah.
But anyway, that's a completely different story. Can't wait to hear these stories, you guys. But let's jump into it. Stem cells. Stem cells are cool. Stem cells are the things that we start with. And stem cells are the things that divide and become just about any type of cell in the body. But stem cells can be found in every type of tissue as kind of a basal layer from which new cells can be born. So there are endothelial stem cells that go on to become new skin cells. Otherwise, if you lost a layer of skin, you'd never grow any back. Fingernails, you have stem cells for that. We even have stem cells specifically to become nerve cells in our brains. So there's been this knowledge of how the fetus, how little babies develop inside the womb. And all organisms, mammalian organisms at this point, or actually all vertebrates, they begin as a little ball of cells, a blastocyst, that then starts to differentiate into different layers. And so we have 3 different layers: the endoderm, the mesoderm, and the ectoderm. And so endoderm goes on to become liver and your gut. It's on the inside, endo. And then the mesoderm is like the middle, but that becomes the heart, muscles, and blood cells. And then you have the ectoderm, or the outside, which becomes the brain and the skin. And so it's always been these germ cell layers. If you have a stem cell that's within the endoderm, it's going to become a cell type that is something that is found within the endoderm. If it's an ectoderm, it's going to become a cell type that is found within the ectoderm, etc. However, some researchers reporting in stem cell reports from Mount Sinai have discovered there is a cell type that changes how all this works. And what they have discovered, and I'm quoting here, one of the researchers says, "We found a stem cell that can become either a liver cell, which is thought to originate in the endoderm, or an endothelial cell that helps to form a blood vessel, which was thought to derive from the mesoderm." So the results go against traditional germ cell theory, germ layer theory, that holds that a stem cell can only go on to become stem cell types within the germ layer that they came from. So this is kind of interesting if we start thinking about how these different stem cell types can become different blood cells. So if you have something like a cancer in the liver, a tumor starts to form in the liver, that means that there's a problem with a stem cell, a dividing cell, a cell type within the endoderm. However, those need blood vessels to feed them, and so there are mesoderm cells that are involved in creating the blood vessels. But in this case, what they think might be happening is that there might be a specific endoderm cell type that can do both. So you can have a tumor cell that also stimulates its own blood vessel development. So you don't have 2 different germ layers that you're dealing with. You're having cells from just within one germ layer, which would affect how you can treat it, because it affects what kind of chemical pathways, signaling pathways are at work within these cells. So it's kind of, yeah, an interesting idea. So now, has germ layer theory been turned on its head? Possibly. I think there are still these definitive layers, but there may be a little bit more crossover between them than what was previously thought. Just kind of interesting. And then additionally, a different stem cell story. Researchers from the NIH, National Institute of Neurological Disorders and Stroke, have discovered that newly formed brain cells in the mouse olfactory system. So your sniffing part of your brain. This is one of those areas of the brain that is fascinating because you have nerve cell endings that end in the nose, receptors that are in the nose, and they go directly to your olfactory bulb. And so the olfactory system is one of the areas of the brain that is directly connected to the outside of the body. We have nerve cell receptors, it seems, for just about every odorant molecule that you could imagine. So there's a massive diversity of these nerve cells. But they found that the stem cells, that newly formed stem cells within the olfactory system, they don't just go on to create new cells to sniff stuff out. What they do is they help to maintain proper connections within the brain, within that area of the brain. So they plugged nostrils in animals and thus disrupted the organization of the olfactory bulb circuits while they were forming. And they showed that this sensory deprivation causes some of the projections in the bulb to spread out, and they lose their really precise conformation that they would normally have. And so you unplug a nostril, and then the olfactory bulb can reconfigure itself and organize itself and become more precise again. So how does that happen? How does this reversal of sensory deprivation happen? Well, it happens possibly because of these new cells that come in, that are born and integrate themselves and can help parts of the brain deal with new sensory information that's coming in during their lifetimes. How do we continue to learn new things? And so these researchers think that this kind of stem cell might be really important in the hippocampus, which is important for memory formation. Help us remember new things for our entire lives.
Yeah, like plop,
Right.
plop, fizz, fizz.
Exactly. So you can remember that because, old cells were there. It got configured into your memory. But then how do you learn a new jingle?
And how do you unlearn an old one? That's the important
You
thing.
don't.
How do I not take perfectly
I think
good
it's called a drill to the
brain
brain.
cells? How do I take perfectly good brain cells that are remembering commercial jingles that I haven't
called
heard
whiskey.
in 20 years? And wait, what is it, Blair?
Whiskey?
Yeah. That's how you fix. That's how you get rid of jingles. Actually, my fear is that I would drink heavily, and all I would have is jingles. I would lose everything else. I'd just have the old jingles. I think they're in there. I think they're more implanted in there than my own experiences in this world.
You'll forget your daughter's birthday, but you'll remember that jingle.
Mm-hmm.
Yeah.
I think that's how it would go.
Then my final story to start out the show is related to different uses for things like nerve cells that you wouldn't expect. Researchers at UC Berkeley have taken proteins from nerve cells, the proteins that create filaments within nerve cells that allow the nerve cells to stay open and conduct along their lengths. They've taken these proteins and actually are using them as a sensitive coating, a surface coating for a new, it's called a what are they calling it? They published it in Nature Communications, and they are creating, it's a biomimetic material using biomolecular engineering and synthetic biology, and they're creating this synthetic coating to keep small particles from clumping together. And so these coatings are often polymer brushes, and they have a bristle-like appearance when they get attached to the surfaces of things. But they've created a biological polymer brush by using these neurofilaments that are that line themselves up on the surface and are a more natural compound as opposed to one that is chemically created. That's interesting. They're taking this protein, this neurofilament protein, out of nerve cells and then re-engineering it and sticking it onto surfaces so that you can have less clumpy surfaces for things like paint and cosmetics. So we could be seeing cosmetics with neurofilaments in them at some point. Your mascara, maybe your eyelashes will be made longer by neurofilaments.
Oh, science, making me so much prettier day by day.
That's right. Your brain can make you sexy. Yeah, it can.
Science. Camouflaging the less attractive women. Is that what?
Or men.
Yeah. Well, I don't know what longer eyelashes is going to do for most men.
I don't
maybe.
judge.
I guess it could be. I guess it's a thing. I don't know.
Yeah, the researcher here is quoted as saying, proteins are generally synthesized with the same exact sequence every time in biology. The length and biochemical order of the protein sequence affects all of its properties, including structure and the ability to bind to other molecules and catalyze biochemical reactions. And this kind of sequence precision is difficult, if not impossible, to achieve in the laboratory using the tools of chemical synthesis, because you have to get all those steps exactly right. And by harnessing the precision of biology and letting the bacterial cell do all the work for us, we were able to control the exact length and sequence of the bristles of our protein brush.
Nice.
Smart brushes from science. This is This Week in Science. Justin, what you got?
So this is in this search for proper antibiotics, and in the quest for probiotics, we may have found, well, it's not really a probiotic, but it's a little combination. We found a robust, broad-spectrum antibiotic. Broad spectrum meaning it can attack lots and lots and lots of things. Robust meaning it's very strong at doing it. And it hasn't been implemented yet, so nobody's really got a good resistance. By nobody, I mean the other biotics, the bad biotics that would be going after, haven't really got a good resistance to it. And it comes from a gene that confers immunity, and the gene that confers immunity to that antibiotic are both found in a bacterium, Staphylococcus epidermidis, strain 115.
So it's related to the Staphylococcus aureus, which is the common source of staph infections.
Yes, which is a whole slew of infections. Now, this particular Staphylococcus creates an antibiotic that I presume it can use to attack other bacteria. At the same time, it has created its own immunity to it, so that it won't be affected by it. Right. So yeah, this is really so they've sort of discovered this. They discovered it on turkeys, is where this was found. And on turkeys, they discovered on turkeys that appeared to have enhanced immunity to bacterial infections. So they studied these turkeys and found, lo and behold, this Staphylococcus epidermis strain 115. It quickly became clear to investigators that strain 115 could produce a potent antibiotic that targets a large number of medically relevant bacteria, including those that cause staph infections, strep throat, severe gastrointestinal diseases, and like. We wanted to know the identity of this antibiotic and the means by which strain 115 protects itself from its own antibiotic's deadly effect. And they did. So they found they've engineered now a version of this strain that was missing this plasmid, that what they were sort of believing, this little it was sort of a clump of DNA that was both seeming to be controlling the antibiotic and the immunity. They removed it, and they found that, lo and behold, the strain 115 no longer produced the antibiotic and was no longer immune to it. So they're zeroing in. This is another one of those sources, like we were talking about this a couple weeks ago, how they were going to be searching more and more for bacteria that's already on skin, that survives there well, to find the next wave of antibiotics, see what's already on the skin, what's already native, and see if there's anything there. And they found one on turkeys now, it looks like. That may be the next wave of potent antibiotics.
Turkeys, who'd have thought?
Right? Another thing to be thankful of this
Well,
fall.
I think that's interesting, since I think turkeys are probably among the animals that receive a lot of antibiotics within the farming or ranching world, the animal agriculture world, poultry, commercial poultry receiving antibiotics. So it'll be interesting to see what they're going to learn from this.
Here's something interesting too. Investigation of strain 115 began as an undergraduate project.
Nice.
And then the bacteria sat in a laboratory freezer for decades.
Oh.
Right. So it was some undergraduate project
That didn't get finished.
apparently didn't get finished, or maybe it got finished, because it might have gotten finished, but you know, it's an undergraduate project. Who actually reads those? At some point, though, they must have gotten looked at before being tossed out and said, "What is this? Oh, well, isn't that interesting? Huh, why don't we just take a look and see what's here?" And yeah, now there's a whole bunch of PhD microbiologists, actual graduate students, analytical biochemists, a whole cadre of people who are working on this now.
Because it could really do some amazing amazing good. People are excited.
I hope they track down whoever did that undergraduate program and find him and like, oh yeah, I went into marketing. It was just a science class I took. I don't know.
That would be great if their name was on the study, even at the very end.
Right.
Just if you could mention them. Because without that undergrad, this wouldn't have happened.
This wouldn't have happened. Now this could go out there and save a bunch of lives. And, you know. Oh.
So great.
Yeah. What comes next?
What comes next? What comes next? Oh. This is kind of interesting. Researchers look into a new way to exploit females' natural resistance to infection. So this is researchers have linked increased resistance to bacterial pneumonia in female mice to an enzyme activated by the female sex hormone estrogen. What is it?
I'm not sure that's quite right.
Is that how you pronounce that?
No, I don't think so.
Females are naturally more resistant to respiratory infections than males. Now, an international team of scientists has shown that increased resistance to bacterial pneumonia in female mice is linked to the enzyme nitric oxide synthase 3, or NAS3. They also show that this enzyme is ultimately activated by the release of the female sex hormone estrogen. The team, led by Professor Lester Kobzik at the Harvard University School of Public Health, introduced Streptococcus pneumoniae into the lungs of mice to mimic the inhalation of bacteria that occurs naturally as we breathe. Female mice and male mice have been treated with estrogen and were able to clear the bacteria from their lungs more rapidly than normal male mice. Female lung host defense cells were also better at killing this and other bacteria than when tested in vitro. Scientists then looked at another set of both male and female mice with deleted genes responsible for the production of NAS3. They found that deleting this gene meant that the female mice were no longer more resistant to infection. The team hoped that in the future this knowledge could be used to enhance resistance to common and serious lung infections. I had no idea.
You said more estrogen. The estrogen kind of helps to...
Good. Estrogen, good.
The antibodies and kill the bacteria is what you're saying.
Yes, more
right?
good.
So my understanding is when you're pregnant, your body releases a whole bunch of estrogen. So wouldn't it be that a big boost of estrogen could be to kill off anything going on when you're trying to grow a baby?
No, so it's really interesting because it's possible that is possible that the mother's body is trying to do that. But what actually happens when a woman is pregnant is that your immune system is not actually as active because the baby is fighting to be able to grow inside you. So it's like a parasite growing inside you and trying not to get rejected. So your immune system actually is kind of it's different.
It's different.
It's different.
See,
it's
because I'm
complicated.
It's this, if it's this two sides of the coin thing, where on one side your body's trying to reduce immune response in your body, but then on the other side, do you see what I'm saying? Maybe it's a give and take to make it just right.
Yes.
I think it's. I don't think it's a coincidence if it's estrogen. We think about when are the main times you get a boost of estrogen in your body. And
bottled water. Oh wait, no, that's me. You?
Yeah. So in a female mammal, usually the biggest boost of estrogen you're ever going to get is when you're pregnant, right?
Right.
So that's immediately where my brain would go. It just seems like but I understand what you're saying. It doesn't exactly compute.
Well, it's not completely immune. I mean, this is talking about it's working in the lungs. This is what they've tested in the lungs to fight off pneumonia infections, lung infections.
So maybe it's only infections in certain areas. Maybe that's possible.
Mm-hmm.
More study needs to be done, clearly. Need to infect those mice with a whole bunch of different kinds of bacteria and see if they all respond to estrogen.
Right. And maybe if there's evidence of certain times of the month, since estrogen does cycle in women on a monthly basis, are women less likely to get infections during the period of the month that their estrogen is high? Are they more likely to get infections when estrogen is low and there are other hormones dominating? Or is there something else at play?
Yeah.
Yeah.
Neat preliminary study, though. Pretty cool.
Yeah. What else you got there, Justin?
What with these fecal transplants?
Yeah.
So we know for a while that Clostridium difficile is a horrible gut disease, which causes hundreds of thousands of hospitalizations, kills more people each year in this country than would die from other causes if those causes are less than 14,000 a year. I couldn't think of anything I couldn't match the number with the 14,000 deaths a year. So it kills a lot of people. This is pretty serious stuff. And the cure that we've discovered for this is fecal transplant. But that's kind of, they got to take some healthy fecal stuff and transplant it into where your fecal stuff should be, or the patient's fecal stuff should be, and it's not easy. It's really not. They have to do a colonoscopy, or there's like a surgery they can do. There's a couple ways they can do this.
Turkey baster.
Mm-hmm.
Oh, goodness, no. Kiki. This is a science show. But now, patients facing Clostridium difficile and the need to do something about it have another option. They can simply take a pill.
Nice.
Which way?
Yeah, which way?
Orally.
Oh.
You can take an oral pill. Yeah, because I don't know how many of them you have to take. And I don't know if I'd be able to take them, quite honestly.
I mean, I'm sure they would be fine. It wouldn't be...
No, sir, you got to think of it like this. You eat the pill and you don't die.
Mm-hmm.
It's fantastic.
Yeah.
If it's that simple.
No, but this is the discovery, though. It's not just that the pill's available now. It's that it's been proven to be just as effective.
That's great.
Just as effective as the more invasive treatments, which whenever you discover that the easier way of doing something is as effective as the really hard and having its own dangers associated to it way of doing things,
so much better.
It improves care.
Yeah, interesting. I mean, what I'm guessing is that they have that the capsules, they're a special material that's going to make it through the acidic environment of the stomach,
Hopefully.
and only open after it gets into the intestine.
Yeah.
Where then it'll open and the small intestine will then be able to be populated. It's past their super acidic environment and it's into the place where all the bacteria want to live.
Mm-hmm. I'm thinking how much cheaper this is going to be than doing a fecal transplant surgery or procedure.
Oh my goodness.
This is so great that you can get that with a pill. So much easier, so much cheaper, so much more available. Doesn't require as much doctor time. It's fantastic.
Mm-hmm. The thing I still don't know from the study is first of all, who puts this into pill form? Who fills the pills? That can't be an easy job. And then the other one is it what I think it is in those pills, or is it something else?
It's probably something else. So they're not just going to take the fecal sample and stuff it into a pill and say, here, eat this. It's going to be a process of making it as not disease-causing as possible. So the people who are going to who are be donating have to be disease-free or pathogen-free. They have to be tested for that. And then they're going to take the sample, they're going to filter it to get rid of stuff that doesn't need to be in there. They'll do special procedures to it, freeze dry it maybe. I don't know. They'll
maybe culture it a little bit.
I don't know if there's culturing involved.
Actually, they are doing freezing. They are freezing, and one of the things they tested freezing, and freezing works. You can do a freezing. And why that was important is that you don't have to do as extensive screening. You get one person who's cleared to be a donor. They can donate more material than needs to be used right away, and it can be stored.
Great. That's awesome.
I wonder how you get on. I wouldn't make that list, I don't think, but
for donating.
Talk about a job that you know you could do. Right? I know I'd be good at this job if this was my job.
I wonder if they have those people on a special diet beforehand. Probably.
Yeah. Oh yeah. No, you don't want to be too. Don't change the diet.
Well, there's going to be.
Tested.
There'll be a diet, like if you have somebody who has a serious problem in their gut, you don't want to add anything, have anything that could cause an allergic reaction and cause any further inflammation, because that would just cause even more problem with leaky gut syndrome and all that kind of stuff. You don't want that to happen. So I'm imagining it must be as. I don't know, what do they probably make you eat like rice and zucchinis before you donate.
Just
right, here's a little bit more of the drill down. The samples that they used in the pill study were filtered, diluted, screened, placed into capsules, and frozen for 4 weeks to enable retesting of donors, original donors, for potential viral infections. So they did the test and they put everything on hold while they continued to test the donor. On 2 consecutive days, each participant received 15 capsules. If their symptoms did not improve within 72 hours, they went through a second course of treatments over 2 days with the same material. Of the 20 study participants, 14 had completed symptom resolution after single treatment and no reoccurrence during the next 8 weeks. The other 6 received a second treatment, and 5 of their symptoms were resolved. So it looks like 19 out of 20 were taken care of within the first few weeks.
Wow, that's pretty fast.
And the one who didn't respond to 2 courses of treatment, whose infection responded to the second course, they had a recurrence. Let's see. So one didn't respond at all. Another one did have a recurrence within 8 weeks. So I maybe call it 18 or 9 out of 10. That's 90%. For the group study. Not a huge population in this study, but maybe not as easy as you might think to get somebody to sign up.
Yes. And if you just tuned in, you are listening to This Week in Science with Dr. Kiki, Justin, and Blair. And do you know what time it is, everyone? It is time.
She loves our creatures, cryback small. 5-peg, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels that are both for her.
All right, let's get right into it, shall we? So, a few weeks ago, maybe a couple of months ago now, I talked about crocodiles and how they were found using tools to hunt for prey. Do you remember this? Maybe a little bit?
Yes, and I also remember they jumped out of trees or something.
That was I think last year we found Crocs in trees. But now, then a couple of months ago, they're using tools to hunt. Well, it gets even more complicated than that. It looks like crocodiles hunt in groups.
Mm-hmm.
And what I love about this is the way these scientists figured this out. So in the University of Tennessee, Knoxville, they recently published this study. The problem is that crocodile behavior is really hard to study. They're ambush hunters, so they're really good hiders. They usually hang out in overgrown waters of tropical rivers and swamps. They're murky. They often hunt at night. And so they're really, really hard to catch in their hunting behavior, also because they don't hunt every single day. They're cold-blooded, so they don't eat every day. So they really wanted to see how these animals hunt, So they decided to reach out to the random person, because it turns out that a lot of people accidentally see crocodiles hunting. A lot of tourists or people that live in the bayou, people just happen across this. Usually it's not a scientist who's sitting there waiting. So the researcher in charge of this, he went to Facebook and other social media sites, and he solicited eyewitness accounts from amateur naturalists, crocodile research, and non-scientists who were around crocodiles. He then also looked through diaries of scientists, and he sat around and did more than 3,000 hours of observations. So with all of this information, he compiled it. Some of the observations in journals that he was looking at dated back to the 19th century, but crocodiles haven't changed all that much in thousands of years, let alone centuries. So he compiled all this, and he found that all of these observations had something in common: coordination and collaboration. So it's not just that they hunted in groups, they collaborated, which means a kind of extra step of working together that's more sophisticated than just a bunch of them hunting at once. So what they found was some crocodiles would actually circle around. They would swim in a circle around a shoal of fish. They'd make the circle tighter and tighter, and then eventually one crocodile would break off and cut across the center of the circle, snatching some fish. Then they'd circle some more, and another one would cut across and catch some fish. So they kind of knew to take turns, which is pretty developmentally intense, right? To know to wait your turn to grab your prey. Then other alligators, large ones, would drive fish from the deeper part of a lake into the shallows where smaller crocodiles or alligators would be waiting. And this is my favorite one. In one case, a huge saltwater crocodile, these things are enormous, they're over 30 feet long, this saltwater crocodile scared a pig into running off of a trail and into a lagoon where two smaller crocodiles were waiting to ambush it.
Are these smaller crocodiles the babies of the larger crocodiles? Why are they sharing with the smaller ones?
Probably, I mean, it's possible, but probably not. They don't really hang out after they're older. They don't stay in family groups. But what's crazy about this is that the three crocodiles knew where each other were. They were out of visual frame of each other, and they anticipated what they would do, and they all had different roles.
Maybe it just means there's crocodiles everywhere in the swamp. Don't go in the water anywhere.
But if I was a crocodile and I wasn't trying to work with these other crocodiles, I wouldn't try to purposefully shoo an animal into another area.
But I don't know. I don't know.
So now, this wasn't actually tested. This is all observational stuff. This is.
Right. And it's observing people who are most likely to observe Crocs, which is swamp fishermen in the middle of where. And look, if we went based on the stories of fishermen, then we have proof of mermaids. There are giant sea serpents, the Kraken, for one, that bring down ships to the bottom of the.
Let's look at this scientifically.
Of evidence possible in science is a visual account.
So let's look at this scientifically. This scientist asked for accounts of crocodile hunting. He did not have any direction. He did not say, see if you can find a crocodile hunting collaboratively. He didn't ask that. He just said, send me your account of seeing a crocodile hunt. And this is over hundreds of years, over a bunch of different continents, from scientists, from fishermen, from crocodile, people that work with crocodiles but aren't scientists. All of these things, all of these different types of people in all these different types of places and all these different time frames.
Yes.
There was one thread for all of these things, and that was crocodiles working together to hunt.
So now that we have what seems to be a bunch of observational data, it's still observational and thus potentially just correlation. Now we need tests. Now there needs to be something done to determine whether they are actually coordinating, because it does appear so.
Mm-hmm.
I don't
Right.
think that this has,
Right.
proven anything beyond a doubt, or,
We need
So
2
we-
pints of water,
we need to disprove the null hypothesis.
about 20
So we
pigs,
definitely need to do this experimentally.
3
But
Crocs.
every time we read a study about someone proving something in a lab, they always say, "Oh, but we're not sure if this is true in the wild."
Exactly. That's
So
true.
you have to do both. However, let me say, I do think there's a little bit of a cold-blooded bias here, and let me explain. There are other animals, like African wild dogs, painted wild dogs, right, that historically have been described as collaborating and having roles within a group and having one dog be the scout and another one to chase an animal into oncoming dogs who are waiting. All these dogs have roles and they hunt collaboratively, and that has never been tested in a lab, but it's pretty much agreed upon that this is what the African wild dogs do.
Maybe because we know that wolves hunt in packs, that this as a relative shouldn't surprise us.
Wolves. It's different. The African wild dogs, I encourage everybody to check it out. There's been specials on TV, there's lots on the internet, I'm sure, about it, but it is different from a wolf pack. Hunting in a pack is different from hunting collaboratively.
Yeah.
So hunting with assigned roles, can only look like a plan.
Oh, but that's what wolves do. Oh, wolves absolutely do that.
It's not the same.
They do
not
that.
as
They
complex.
have chasers. I mean, they have some that are chasers, and some that are supposed to just go around and head you back into where they're waiting and conserving energy to put on the kill. They do all these things. But go ahead.
What I'm saying is, in the studies of the wild dogs, it's not the same as pack hunting in wolves. It is more complex. Animals have designated roles. It's possible that when wolves hunt, some of them peel off, but it's not always the same ones. It's not the same ones that have designated roles that they say, this is what I do. When you watch African wild dogs, it's almost like they have a huddle, and then they go off and hunt, and it's this very delicate, it looks planned out, exactly what a wild dog does. And biologists are convinced that it is some of the most complex hunting in a group of any animal we've ever seen. And there have never been needs to do studies in a lab on these guys. You see it and you believe it. And I think that's for 2 reasons. One is that they're warm-blooded, and so we think they're capable of more cerebral behavior. And 2 is that it's on land. And so we can see it.
Watch it.
We can see it in a helicopter. We can see it from beginning to end. We know exactly what's going on. There is no doubt of what's going on in this group of wild dogs. But with the crocodiles, they're this reptilian thing that is not supposed to be very smart, and a lot of things happen under the water. And so people don't really know how to classify them, and they don't really want to...
I don't know if we... I don't know. Yes, maybe we have always thought of them as not very smart, but I think it's more that we think of them as ancient, ancestral, so we think of them as stuck in an older form, and therefore they wouldn't have as many advanced behaviors as animals today.
Yeah.
I think that's more of like where that idea would come from, as opposed to, that it's a cold-blooded bias.
That's certainly possible. But I think that you could ask a random person on the street, what's smarter, name a mammal and name a reptile, and they're going to tell you it's the mammal every single time.
Oh, sure. Yeah, with birds, I mean, we still have the whole term bird brain.
Yeah.
It's not a cold-blooded thing, but it's not mammal.
Yeah. I just wish then, if it is about them being so old, one thing I do wish I could do is get in a time machine and see if crocodiles were doing this 1,000,000 years ago. Because it's possible that physically they're fine. They don't need to evolve anymore. Physically, they're done. So there's no physical evolution going on for that long. But maybe their brains are still changing.
Mm-hmm.
Right. We are all equally as old. This is just to make that point real quick, even if we haven't had to alter or
The
have
point
had
is,
to
you
alter.
could go back to when the only mammals were little shrew things living in the ground, and you would see a crocodile that looks pretty much exactly like a crocodile you see.
Yes.
Yes.
Yes,
Yes.
but they've still had
They're
plenty of
older.
time on the planet to need to adapt and adjust and do things like you were pointing out.
Yeah.
And we've seen it with dolphins. We've seen dolphins, and we've seen the orcas working in groups. We've seen all that sort of group hunting stuff going on, collaboration. I guess I wouldn't. I don't have a problem with it. I wouldn't mind believing it. I just don't know that. The idea, the excuse that they're in a swamp and therefore we can never see it, so we've got to take a bunch of third-hand accounts of if they can. I don't know.
Well, it's
Go
a
out
start.
there and videotape a group hunt. Get it on film. That's the only way we believe it.
I'm sure you can. I'm sure there's films of crocodiles
I'm
hunting
not
all over
me.
the internet.
Not me.
I wonder if Wild Kingdom or any of the David Attenborough documentaries, if they went out and accidentally filmed something but didn't actually think they had seen
Exactly
what they got on film. So maybe if people go back and look at, swamp documentaries, maybe we'll find something there.
Totally, definitely. And Justin, I just want to say we have to be careful how we use certain words. And so when you say that dolphins hunt collaboratively, it's not the same. What we've seen dolphins do is not the same as what I was describing with the crocodiles. So it's a whole different level.
Well, what I was describing with the dolphins, though, is when they can work together to create a wave that knocks a bunch of fish up onto a shore. These are river dolphins, the one that I was thinking of in this particular one.
Right.
Where they work together and they have to all face the same direction and hit the shore together, and they put a big wave out that puts the fish up onto the shore. Then they grab a couple of them and wiggle back out. But they have to work as a coordinated team in order to enact that maneuver.
So there's a difference between me saying, let's work together to make this souffle, right? And we all put in equal time on equal parts. You whisk a little bit, I whisk a little bit. You watch it for a while in the oven, I watch it for a while in the oven. That's us working together to make a souffle. But us collaborating to make a souffle is me saying, you're a really good whisker. You do the whisking. I'm really good at knowing exactly when that souffle is done, so I'm going to take care of the baking. That's kind of the difference.
And and thinking about, you've had some other stories related to kind of manipulation, where one animal, where two animals of different species, I think it was the two different fish species, or the fish and the eel.
Yeah.
And so the fish would chase the prey into the reef where the eel was staying, and then the eel would either get to eat it or would chase it out or fight. I don't remember, or the other way around.
Mostly, yeah, that was it, yeah.
Yeah, and so there's the question, it's cooperative, but is it manipulation?
Well, in that
one species of the other. So it's not necessarily collaborative.
Was actually gesturing, and would select eels that were better at the task of chasing them out.
Yes, but the eel sometimes got food,
and
but when it didn't, it had no benefit to doing it. So
you got told where to go look for food, at least.
So it was definitely a case of manipulation. The eel wasn't entering into it with the same understanding of what was going on as the fish.
Yeah.
So there's these many complex layers of group hunting that's really where we're trying to make this definition of what is more complex, right? And so that's where this comes from.
And I just found a paper from 2012 published in Behavioral Ecology and Sociobiology, Group Hunting Within the Carnivora, Physiological, Cognitive, and Environmental Influences on Strategy and Cooperation. And they have a bunch of types of hunting that might come in handy, terms and definitions that might come in handy for this. There's passive, Similarity, the old definition back from 1989 is that all hunters concentrate similar actions on the same prey, but without any spatial or time relation between them. At least two hunters always act simultaneously, and similar actions are varying elements of pursuing a prey, like stalk or chase. And then now, timing and orientation may primarily be in response to the behavior of the prey and not that of other hunters, and the kill is shared. There's synchrony also. Each hunter concentrates in similar actions on the same prey and tries to relate in time to each other's actions. And they may begin at the same time or adjust their speed to remain in time. They synchronize the timing of their movements in relation both to each other and to the prey.
Okay.
Then there's coordination. Each hunter concentrates in similar actions on the same prey and tries to relate in time and space. Hunters can begin from different directions or adjust their position to remain coordinated. And that positioning relative to one another is not incidental. For example, if prey are approached from different directions, individuals must have spread out at some point to positions that allow such an approach, rather than be individuals arriving at the scene from different start points. Let's see, for example, fanning out along a front to flush prey or encircling prey such that all individuals have a similar role. The prey is shared. And then there's collaboration. Hunters perform different complementary actions, all directed towards the same prey. Examples are driving, blocking escape way, and encirclement. And there must be a clear role differentiation resulting in team-like behavior. The same individuals may perform the same specialized roles in different hunts, and timing and position are more strongly based on each other rather than the prey's behavior than during lower levels of cooperation.
Nice.
Yeah, so we got some definitions there.
That's perfect. Yeah.
Yeah.
So you can see from what I was talking about with the crocodiles, we saw a few different levels in the accounts that I was talking about with the crocodiles. So
yeah, many different levels. So it'll be interesting to get into that a bit more.
Yeah, exactly. Cool. So, sorry, we went way off on that one.
No, this is good. It
was a fun conversation. That was good.
Yeah. So then I have a couple real short ones. For example, that birds, when they're in turbulence, when they're in a super windy time when they're flying, they actually collapse their wings and engage in free fall because keeping their wings open is too difficult, and it's kind of actually working against them to try to keep their wings open. So they close up their wings when there's high turbulence and fall. And they can do that up to 3 times a minute when there's really turbulent winds. And so they're saying that this is the same conditions where many airplanes, smaller airplanes, would be grounded. Then these guys can stay up because they're doing this. And so they're saying that it has potential for application into aircrafts, which is terrifying to me.
Right?
Terrifying.
Ladies and gentlemen, we're going to be bringing the wings in for just a little bit, do a little bit of a free fall, clear this turbulence, and we're going to put the wings back and be on our way again.
Yeah, terrifying.
No.
I want no part of that.
Did he just? How'd I heard him say? Because I don't know.
Yeah, terrifying. And then the last one, gecko feet. We've gone on and on about gecko feet in the past. We know a lot about them now. They have these setae that they actually have to curl their toes as they place their foot on the surface and uncurl them to have them stick, and then they curl them back up to release. And they're directional. They only work when they're going up this way. And for the first time now, they've studied when geckos go downhill. They found that geckos actually swivel their wrists.
Aha!
Yeah. So on a 45-degree downhill slope, the geckos rotated their hind limbs up to 70 degrees toward the tail.
So there's no magic to how the molecular forces actually work, because everybody's been like, well, if you put your feet on this way and you pull them off this way, if you're going down, aren't you just going to fall off?
You would, but
you would, but
rotate.
All they do is turn their hands. I love that.
Yeah, fantastic. So that's the latest in geckos, and that's all I got for the animal corner. I think it's plenty.
I think that was wonderful. Thank you so much for an informative and inspiring Animal Corner. Everybody, this is This Week in Science. It's time for us to take a little break, and we'll be back in just a couple of moments with more science. You know what we've got? We've got Rigs versus Reefs. We've got Kanga-Shoes and Higzogenesis coming up in the second half, so I hope you feel like staying.
Explain the things you heard with more than intuition.
Hey everyone, I just want to say thanks for supporting TWIS. We have made it to our first milestone on Patreon, $500 an episode, all thanks to you. We're going to keep going and keep striving, though, trying to reach ever higher amounts of support so that we can make more science content for you. The more support we have, the better able we are to take time and equipment to be able to create content and put it out there for you. So really, you're helping yourself out by helping us. And if you want to help us out some more, if you haven't helped us on Patreon before, you can head over to patreon.com/thisweekinscience. Patreon is Patreon. It's kind of like Kickstarter, where you support us and for every episode that we produce, and then we'll send you a little gift thanking you for your support. The level of support determines the little gift that you get. You can donate any amount, $2 per episode, $20 per episode, whatever you feel like giving, but it does help out in the end. There is a giant spider! Ah! It just came from the sky! I'm glad it didn't land on my head. Oh my god, sorry, I just freaked out, everybody. It is behind my monitor. Ah! You're big! It's like that big! Okay, sorry. Spider.
Do you need to take care of the spider and I can finish this?
I just got goosebumps all over myself. Okay, it's not after me. I am fine. I am not in the movie Arachnophobia. We are okay. Anyway, any amount of support that you are able to give, we really appreciate. And like I said, it just helps us do everything for you. So it helps us host stuff, it helps us hire contractors, it helps us buy equipment, it helps us do all sorts of things. If you don't like Patreon or that whole idea, you can sponsor us using PayPal. We have buttons all over our website for donations through PayPal, and that website is twis.org, TWIS.org. If you would like to support us in a different way, like maybe wearing a bag or a shirt or a hat that has the TWIS logo all over it, you can buy our merchandise. TWIS.org has a link to our Zazzle store, zazzle.com/thisweekinscience. You can find all sorts of TWIS emblazoned swag for you, for a friend, for a family member, just in time for the holidays, folks. I don't know, is there a TWIS Halloween costume? No, we don't have a TWIS Halloween costume. But anyway, you can look for those fabulous things there on the TWIS website. And if financial support is not something you're in a position to give, we would love your help just trying to find new listeners for TWIS, because the more people that we can get listening, maybe the more people will be curiouser about the world. And I don't know. It's a big, fun, TWIS happy family here. So if you can help us out by telling people about TWIS and our weekly broadcasts this month in October on Wednesday evenings at 8 p.m. Pacific Daylight Time, we would really appreciate it. Use your social networks for science. Help TWIS out. We really appreciate your support, and we could not do this without you.
explain the things you heard with more than intuition. A line of reason shows the way to go. No conclusion. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't see.
And we are back with more This Week in Science.
Oh, yeah, we are back with more science. Justin, what other stories do you have to share?
All right, let's see. What do I got queued up here? I have got, ooh, yes, delisting the drought.
Yeah, so this isn't the biggest drought in California history?
Apparently it is not. No, apparently that's the other. If it was, then the premise of my bit here is going to be lost. So this is a, let's see, where is this coming from? I've got this from the American Geophysical Union. They're saying the 1934 drought was by far the most intense and far-reaching drought of the last 1,000 years in North America. And they put the cause in part due to atmospheric phenomenon that may have accelerated the drought in California. I believe this is, if I'm not mistaken, the Dust Bowl?
The Dust Bowl.
I think so. So, for the first time, much of the heartland of America had been farmed and tilled, that sort of thing. We ran into a drought, crops were dying, and the wind came and was creating giant windstorms because you had all this tilled soil on the top that was all nice and easy and ready to be brought up into the atmosphere. This may have been part of the acceleration of this drought. Yeah, they find that the extent of the 1934 drought was approximately 7 times larger than droughts of comparable intensity that struck North America between 1000 AD and 2005. Nearly 30% worse than the most extensive severe drought that struck about 1580. So, and this is one of those things I do sometimes say this whenever we are running into a drought situation, that we've been in a wet spell. You know,
In California. Yeah.
I mean, California's had a wet spell. That's sort of 1848. We found it in good times. We got hit once, really, reality check in 1934 there. Another time, 1976, where we had a 2-year dry spell, which was often considered the worst drought in the state's history. We're getting there now. The drought that we've got currently is a 3-year drought. It's considered to be going to continue throughout 2015 as well. So they're not really looking for this drought to end until sometime 2016 into the 2015 season, I suppose.
Well, it's good to know. It's good to know because we'll probably be able to get through it and we'll probably bounce back, which will be nice. But I still feel like you're kind of saying, well, I was stabbed, but I wasn't stabbed as badly as the last time. Okay.
Well, I've been stabbed worse before.
I was stabbed worse before. Well, it was in my arm, but now I got stabbed in the throat. It's not as bad, but it'll be fine because this is our bread bowl. This is not good that we're having a drought where we create the majority of our food, where we grow the majority of our food. Whether it's the worst drought we've ever had or not, it's having a huge impact on our lives.
Okay. That's a good point. Here's the other side of it, though. Here's the other side of it. This is an economic issue, not a water resource issue. It's an economic resource issue. And it's largely because we do export a tremendous amount of produce out of the country and around the country from California. So when you're saying that California is in a drought, and it's not really the worst drought we've had, what you're also saying is that we're managing our crops perhaps not as efficiently as we can. Because if we're in a drought, and our main export is produce, then you really got to backtrack a little bit to where that resource that creates the produce is, and you say, in times of drought, should California be exporting water? Because that's essentially connecting the dots. Our major export in this state is water, and yet we're in a drought. So there needs to be some balance there, and that's what I'm talking about. That's what I keep harping on, is that the agricultural processes that we use need to be refined, and that's where the intense focus needs to be placed on. If we're going to continue to export water, we need to manage that water we have better, and it's going to come down to agriculture.
Yeah, well, one of the biggest things we export is almonds. And
those almonds you've got there.
Oh yeah,
Are those California grown almonds? California grown
they're
almonds?
not actually almonds; they're almonds.
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 Use
What I've got here in this can here, these are almonds. Now, when they grow on the tree, they're called almonds. But the way the farmers get them out of the tree is they shake the L out of them, and so what you're left with is almonds. This is
funny.
Oh, I just put into Google how much water does it, and the first result was take to grow an almond.
Yeah.
A lot.
It's a lot. I want to find the number, but it's one of the water greediest crops you can have, and I think it's our #1 export in California is almonds.
It's up there. It's we're the #1 producer of it. I don't know that it's our #1 export, but we are the #1 producer of it.
99% of all U.S. almonds come from California.
Yeah. I don't know that means it's our #1 export, but it does mean we're the #1 producer.
Avocados are another large water-greedy, water-gulping
Yeah,
crop.
but they tend to actually only grow in a place that still gets wet. That's the nice thing about them. I mean, they're not... They're
talking about San Diego?
No, yeah, well, San Luis Obispo.
So let me
Santa
give
Barbara.
you some water facts. Are you ready for this? One almond takes 1.1 gallons of water to make.
How many gallons of water? That's larger than, like, how many showers of yours, Justin?
It was this or a swimming pool. I had to choose.
Yeah. One head of broccoli, 5.4 gallons of water.
Right.
Man, I love broccoli.
One walnut, a single walnut, 4.9 gallons of water.
Exactly. So when we talk about being in a drought, when we talk about not watering lawns, when we talk about this, and yet you're in an ag state that hasn't really updated the way it uses water. Now we've updated crop rotations, we've increased them, so we've actually got more crops growing on the same land than we ever have before. We've improved the way that we get it. We don't really fill stuff with silos anymore because now our transportation system is such that stuff goes from farm and gets shipped almost immediately. We've improved the from farm to table aspect of this business, but there hasn't been. We're still doing irrigation. We're still doing run the water to it, run the water down the thing. You look out, you drive these farm fields, you can still see the giant sprinklers that are evaporating a good percentage of the water before it even gets to the crop. So there are things that we can do. There are things that need to be focused on, and it should be a major works project that we do instead of just worrying about running more water from one part of the state to the other. Where it's ending up, how it's getting used, needs to be the focus. That's where resources, time, and engineering needs to be applied.
You know what, too? That would help the economy and provide jobs.
What?
What?
Both? Improve the economy, protect the water supply, and wow, I could have a green lawn, too?
This is crazy talk.
Right? I know.
Yeah. Do you know what other crazy stuff is happening?
What?
Talking about things that are having results that you didn't expect. California's most productive fisheries are offshore oil rigs. Not coral reefs. It turns out, based on an analysis, researchers knew that there were a lot of fish kind of hanging out around the ecosystem that oil rigs create offshore when we stick these massive things, have them float on the ocean, and have steel beams coming down and have them connected to the floor of the ocean. It creates actually a lot of potential ecosystem, and researchers took a look at the productivity around them and found they used an annual survey performed with a remotely operated vehicle. Different sites were visited for at least 5 years, some as many as 15, and they looked at fish abundance and the diversity of species by visual assessment, like looked at them and said, that's one of those, check. They estimated total biomass, and the change in biomass between years was then normalized to the area of the seafloor that was covered by the survey. They found the most productive places in the literature, a reef in Tahiti and an estuary in Louisiana, saw about 75 grams of productivity for each square meter. The lowest of the oil rigs came in at 105 grams per square meter,
Wow.
and the highest, almost as high as 900.
Holy smokes!
Yeah.
Right.
And so they were being conservative in this because they were only using fish that were found within a couple of meters of the structure. So it doesn't take into account the entire water column that is enclosed by the legs of the rigs. So there may even be more productivity. What they're saying is that the fact that you have the oil rig extending from the surface to these really significant depths, that maybe what's happening is as you follow it down, fish could be attracted to it because they can spend different parts of their life stages basically in the same habitat, just moving through it vertically. Yeah, but maybe it allows fish that like multiple depths to find a really neat place to live and enjoy it, but they think the vertical structure is what contributes to this, but they have no idea.
Yeah, that's what I was going to say. It sounds like an artificial reef.
Yes, it's like an artificial. These offshore oil rigs are like artificial reefs, and now they're wondering, as we're building things like wind farms, and as some of these oil rigs, maybe we decide to take them offline because they're not producing as much oil anymore, should we leave them? And should we actually keep them where they are because they create this habitat that might actually be very beneficial to fish and to the oceans?
Could we sell one to a private party who would then turn it into a Science Island Sea Lab?
Could we? These are all questions we
I'll
don't
work at
know.
the Science Island Sea Lab.
That sounds pretty awesome, doesn't it?
I like that. Science Island Sea Lab. Let's do it. Yes. Do you have another story, Justin?
Oh, yeah. This is, okay, so remember the giant, I don't know if you remember the giant kangaroos? 8 foot, 550-pound kangaroos that used to be in Australia?
Okay.
Oh, yeah. They're
Huh?
big carnivorous kangaroos.
Big carnivorous roos.
Yeah.
This is surprisingly scientific research based in Australia, which you don't see that a whole lot. Okay, so this is, I like the way it starts. Imagine that a time machine has transported you to the Australian outback 100,000 years ago. As you emerge, things look pretty much as they do today. No, it doesn't see that. It says, you see a huge kangaroo with a round rabbit-like face foraging in tall brush nearby. The animal's surprising size makes you gasp aloud, but when it hears you, becoming equally unnerved, it runs away. Doesn't hop away, mind you. It runs away on all fours. Or no, it doesn't hop away. Yeah, so this is, no, it says it walks on its hind feet. It walks. It bipedally walks away. So if you've ever seen a kangaroo, kangaroos kind of hop, but they also do like sort of like front paw. Yeah, they also sort of run. They're sort of like
you've ever seen a mouse run, they kind of look like mice, like giant mice. Because mice have nice big hoppy back feet, but they still use their front paws, right? Kangaroos are sort of like that. They're just giant mice. They can hop, but they equally use the front paws for locomotion. But this 550-pounder, based on studying the bones, they found there's stiffness in the spine, some other places, but they also just the physics of a 550-pound creature hopping was troubling. But yeah, after doing a more thorough investigation, they're saying it looks like it was likely bipedal and walked left, right, left, right, left, right. And this is an amazing looking creature. It really does. If you just sort of, it looks like a T-Rex with fur and a rabbit head.
That sounds a little disturbing.
Isn't it?
What's interesting is the kangaroos of today, they don't have kneecaps, so they couldn't walk. They're incapable of bending their leg in a way to walk. It's stuck always bent like that so that they can hop. But yeah, they don't have kneecaps, so they can't walk. So that means that since then, that means they had kneecaps, they lost them so that they could be better at hopping. So what makes hopping so much better than walking, I wonder?
Maybe once you got small enough and you had those big powerful legs still from back when you were 550 pounds, or the ability to have them, maybe it was just that combination allowed it. One of the things they're looking at is the ankles. In running animals such as horses and dogs, the lower end of the tibia has a flange that wraps over the back of the joint, providing extra stability to support more weight on each ankle. Yeah. Living kangaroos almost always distribute their weight equally over both feet, don't have that flange.
So maybe the flange was lost?
The flange must have been lost because
what a weird looking critter.
The ancient roos, 8-foot kangaroos, had bigger hips and knee joints. Knee joints, it says. Shape of the pelvis differs importantly as well. Giant roos had a broad and flared pelvis that would have allowed proportionally much more gluteal muscles than other kangaroos. Those muscles would allow them to balance weight over just one leg at a time. As do humans when we walk.
What a weird critter.
Yeah, and their name, the Sthenurines, Sthenurines.
Say that 5 times fast.
Sthenurines.
Yeah. That's what I'm gonna name my firstborn now. I've decided. Sthenurines.
Sthenurine.
So yeah, it's a really bizarre. Kiki, you had the picture up. That was brilliant. It's such a bizarre looking, because you see that and it does, right?
How bizarre! How bizarre!
Bunny rabbit with a kangaroo body that walked, kind of looked like with the little arms like T-Rex. He was a carnivore. Was definitely a carnivore. And the picture belies the fact that you've got to have it in. You've got to put a person there to relate to the size, because this thing is 8 feet tall, 550 pounds. It's a beast. It is a beast.
It's probably big and fast on 2 legs and
kind of cute.
I would have
terrifying.
Terrifying. Oh my gosh. Let's see. My final stories for tonight. Hygzogenesis.
Say what?
Hygzogenesis. Yeah, researchers have been trying to figure out how we ended up with this asymmetrical universe where we're full of matter, right? Where did all the matter come from when antimatter and matter are supposed to annihilate each other? And if you start out with the same amount of matter and antimatter, how do you end up with more matter dominating? It doesn't make any sense. And so there's a paper that's in Physical Review Letters, and it's outlining a new cosmological model that is called Hygzogenesis. Hygzogenesis refers to when the Higgs particles started appearing in the universe. So at one point, there was just heat and stuff, and then the matter, things that made matter had to come before matter, right? So if you have Higgs particles that are supposed to carry the mass for the matter, then, and matter is like related to the baryonic protons and neutrons, baryogenesis, you're going to have Higgsogenesis happening at some point even before that, but we don't exactly know. So it's hypothetical. This is just a hypothetical cosmological model. And what they think, they propose that the Higgs has an antiparticle called the anti-Higgs. And so the proton has an antiproton, and the electron has an antiparticle called the positron. And so these things, regular matter stuff, appear in equal amounts. And so where did the unequal stuff come from? They think that there was an asymmetry in the beginning between the Higgs and the anti-Higgs, and that is what gave rise to matter dominating in our universe. They suggest that the Higgs particles could decay into dark matter particles, and that's what would have led to dark matter. And so now this would lead to potential experimental methods to look for Higgs, the decay of Higgs particles that would indicate dark matter or an interaction with dark matter. So we might be seeing experiments, or we might be looking at data from someplace like the Large Hadron Collider as it gets more and more powerful, to be able to actually see whether or not there's something to this hypothetical idea. There will be more papers coming on Higgsogenesis.
Nice.
The only thing I'm afraid of, though, is that we're going to drill down this, and we're going to find out that matter didn't win out over antimatter, and in fact, we are actually the antimatter, and we've been living in the wrong universe this whole time.
Durr. Ow, why'd you make my brain hurt?
It's actually,
Oh.
there is no, I don't think, at least, that there is any way, I mean, you know, after the fact, we've defined this as the matter, the other one as the antimatter. But I think we could just as easily be living in the antimatter world as the matter world, and have the antimatter world having one out, because it wouldn't make a difference. But
It's just based on what we call it here in our own universe. There still is.
Point is,
Yeah.
we needed the symmetry part. That was important. Didn't matter which one, which side won. We're here now.
Yeah. Oh, and then astrophysicists from the University of Western Australia at the International Center for Radio Astronomy Research have been trying to probe for dark matter. They're trying to figure out the weight of dark matter in our galaxy. They used a method that was developed about 100 years ago, and they found that the weight of dark matter, that by looking at the edges of our galaxy, the Milky Way galaxy, looking at the edge of it, they found that the weight of dark matter in our galaxy is 800 billion times the mass of the sun. 800 billion times the mass of the sun. But they say it's about half as much as was expected. So they've measured, and there's less dark matter than was thought. So this could kind of put a wrench in some further dark matter developments.
Still a number so large that my feeble human mind can't even begin to comprehend it.
Yes. Oh, and also they were looking at things like, well, if you know the mass of the universe, of the galaxy, then you can figure out how much momentum is needed to be able to hit escape velocity. And so you need 11 kilometers per second to leave the surface of the Earth in a rocket. You need to hit 550 kilometers per second to leave our galaxy. Now we know the galaxy's escape velocity. Just kind of fun. Kind of fun. Any other stories out there, you guys?
Sure, I have one. Holding it. We've all done it.
You mean like urine?
Yes. Yes.
Dang it.
So scientists have been studying which neurons are responsible for it, not only when you do it intentionally, but just the unintentional background of always holding it. No matter how much or little you have in there, you're always holding it. And so scientists are trying to find the specific regions of the brain and the specific neurons working, and they found that there's other muscle groups that are related, so they're trying to find a map through that. So if you squeeze your glutes, you're also squeezing your pelvic floor. If you contract your toes, there's also a little bit of a contraction on your pelvic floor. But if you wiggle your fingers, there's nothing. So there's certain muscle groups in your body that are related in terms of neurons to your pelvic floor muscles, which are directly related to holding in urine and not wetting yourself. And so they're just starting this map so that they'll be able to help people who have problems with that, especially it's mostly women and older women that have problems with that. So it could potentially help with that quite a bit. So it's pretty interesting that they're just now starting to do this map and that they're related to other muscle groups.
So potentially you might add toe scrunching to your Kegel exercises.
Yeah.
Well, you know, I actually it's not that surprising because I can tell when my kids need to go pee.
Because of the dance they do?
It's the dance, but it's also like you can see all of a sudden there's like a bouncy leg thing going on. And you go, go pee. I don't have to. Yeah, you do. No. Okay. Minute later, running for the bathroom, right? Here's the one I forgot. The way you walk can affect your mood.
Oh, do tell. Does that have to do with posture?
Slump-shouldered when we're sad, bouncing along when we're happy. Researchers have shown it works the other way around too, making people imitate a happy or sad way of walking actually affects their mood. So in this study, they did basically a memory study that people look at a list of depressive words and a list of positive words. And then they had them walk in a highly instructed way. One was to, of course, simulate a depressed walk. One was to simulate a happy walk. At the end of the walk, they gave them a pop quiz on the earlier vocab. Those who had walked in a depressed style remembered much more of the depressive words from the earlier list. Those who had walked in the happy way remembered more of the positive words from the vocab earlier, suggesting that the way people were walking was affecting their mood.
Nice.
Whistle while you walk. There we go. Is that it? Are we good? Do we have one more?
Why don't you give me an Ebola update?
Oh yeah. The Ebola update.
So my prediction was wrong. I predicted that there would be not one more new case, one case in the United States. Then sure enough, there's a case, somebody dies, and somebody else is infected. There is a parameter. I should have done this disclaimer. Anytime I'm saying something can't or won't happen, it is not meant to include the states of Texas, Florida, or Ohio. Those are exempt from my predictions. That should just be a given. Anything goes in Texas, Florida, or Ohio. I can't predict those places.
Okay, so it is location specific when it comes to Justin's predictions.
It's anywhere at all in this great land of ours except for Texas, Florida, and Ohio.
All right. Ebola update for this week. We have 2 nurses who are infected with, who have been tested and shown to be infected with, have contracted the Ebola virus. There are questions currently as to the procedures that they used, whether or not they did exactly follow protocols for safety, whether or not there were problems with the protocols. So a thorough review of that is underway. One of the nurses had just taken a plane flight like a day or two before being confirmed as having contracted the virus. EbolaDeeply.org is a new nonprofit organization, a journalistic organization that has been started in order to educate people and keep people around the world up to date on what is going on with the Ebola outbreak. I highly recommend it as a source of information, as it compiles news from sources around the web and also publishes articles and summaries of things that are happening. They have an Ebola executive summary to give an overview of what's happening in the Ebola outbreak, latest developments curated. There is a case map so that you can see, get an idea of the numbers of people who are infected in various parts of the world, specifically Africa. The data is sourced from the World Health Organization. It's a great website, great resource for people who are interested in the news about Ebola. So far, there are 8,401 total cases of Ebola that have been reported, according to World Health Organization data. And that is where we currently stand. And at this point, for people who are concerned and having conversations about Ebola and your risk for Ebola, it is still much wiser for you to go out and get a flu vaccine and to worry about something like the flu than it is for you to worry about Ebola.
Hear, hear.
Unless you live in Texas, Florida, or Ohio.
No.
In which case, your biggest concern should be living in Texas, Florida, or Ohio.
Ouch.
Okay. All right. Ouch. Seriously. Oh, dear. Oh, dear. Flipping out, sewing. Are we good? Are you gonna?
Oh yeah. No, the sewing thing was funny, and I don't know. I think it's an economic question. I don't even think it's a science question. But they did a study. They found that the millenniums don't know how to sew, don't know how to put a button on, don't have a needle and thread in their home. When a piece of clothing breaks, they simply throw it away.
Wow.
Now, I think I'm on the border there a little bit in that region between, because my grandparents all had sewing kits, big sewing kits. They mended socks back in those days. I know I have never mended a sock. I've never put a sock back together. If a sock gets a hole, it's done. That's the end of the line. But a button comes off, I'll put a button on. I have some sort of threshold. Apparently in the millenniums, there's a new generation coming up. Clothing breaks, you throw it away, you get new clothing. Part of that, I think, though, why I think it's an economic issue, not just a lack of education and home economics. Clothing, I think, is probably much more affordable today than it was in our grandparents' era. And as we've moved that way, it is becoming easier and easier simply to throw it away. And even though we may have a glut of clothing in landfills, it is taking up space, but it's not one of those things that you look at and go, oh, that clothing, that jacket is going to be there for like 3 years before it's decayed, or 5. It's not. It doesn't rise to a level that we're too objective.
Not to mention the stuff getting made in a factory. A lot of it is burning so much fossil fuel to run the machines that are making this clothing, and then a lot of it's getting made in Indonesia or China or India, and then it's getting shipped here.
Over.
It has a huge impact when we buy new clothes all the time on our carbon footprint. It's definitely something that people, you're right, they don't connect the dots. They think about everything else in their life as it relates to consumption and fossil fuels and all this kind of stuff, but they don't think about it about their clothes at all.
Yep. Last story too. Earth's magnetic field for years and years. We're kind of due for one. It should be coming, but it's presumably the magnetic field starts to get unstable, and it slowly goes through this process where it's going to flip, and it takes some thousands of years to reverse directions. Maybe not so much so. It's now being determined by researchers at Columbia University, University of Berkeley, that this process can and maybe did happen in the past within a less than 100-year span of the Earth's magnetic field becoming unstable. So it's something that could happen within a human lifetime.
Interesting. Well, it'd be fun to see if that actually happens. That we've been reporting on the magnetic field flipping for years, and it would be really cool to actually.
I don't even know if it would. I think it would mean that the toilets would flush the other direction.
Whirlpools would go the other direction, right? No.
And that would be the big difference. And you'd have to repaint all of the compasses, or you just change the maps. I don't know what's easier. Change the maps to call the south north and the north south.
You don't have to, because it's all arbitrary anyway.
Right? It's all arbitrary.
What?
Down on maps, on GPSs, on compasses. We just have to figure out a new way of doing things. That would be the big adventure.
Yeah. Well, I think we have hit the end of our show, everybody. It has been a big adventure. This Week in Science would like to thank everyone who is supporting us on Patreon. Thank you for helping us reach our first milestone of $500 an episode. Yes. Please help keep us here. Please help keep us growing to our next milestone of $1,000 an episode. Help us get to the point where we can start doing some really great work for you guys, making some cool science content and really bringing you the science. So thank you for your support. Brian Condren, Kevin Donald Wesley Ballard, Paul Disney, Sam Winsier, John Ratnaswamy, Rudy Garcia, Steve Dabell, Aubrey Burgess, Stephen B., Jason Dozier, Matthew Litwin, Eric Knapp, Andreas Akestad, Jason Roberts, Patrick Cohn, Shane and Tara Ginsberg, Byron Lee, E.O., Bob Calder, Jared Lysette, Ulysses Adkins, Jake Jones, Brian Hedrick, Cassie Lester, Chris Cross, Sarah Chavis, Stephen Sorowiak, Layla, Paul Stanton, Marshall Clark, Charlene Davidson, Henry, Violated Gorilla, Don Kamarechka, Randy Mazuka, Ed Dyer, and Tony Steele, Lord Trentoni and Moody, Larry Garcia, Richard Bowersox, Kevin Parachan, Kurt Stefan, Michael George, Craig Landon, Russell Jensen, Mountain Sloth, Jim Drapeau, Darren Lute, Tara Payne, Alex Wilson, Daryl Lambert, John Maloney, Jason Olds, Gordon Grant, James, Paul West, Lauren Lang, Ed Bennett, Thomas Mikinen, Alec Doty, Illumina Lama, Nick Gradwell, Dan Rambo, Dougal Campbell, TMRO, Miko Pakala, Craig Porter, Adam Mishkan, Jason Martin, Aaron Luthan, Marjorie, Scott Lajewski, Tyler Harrison, Ben Rothig, Columbo Ahmed, and Gary Swinsberg. We couldn't have gotten here without you, and we can't go further without you. You make TWIS happen. Next week's show, we're going to be broadcasting live Wednesday night, 8 p.m. Pacific time at twis.org/live, where you can watch and join our chat room. But if you can't make it live, don't worry about it. You can always catch the podcast or the video later, twis.org, or youtube.com/thisweekinscience. And if you would like to support us, you can find information for Patreon at patreon.com/thisweekinscience. And thanks for telling a friend about TWIS.
And thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory, or if you have a mobile device, you can look for TWIS for Droid app in the Android marketplace, TWIS in the Apple marketplace, or in the new iPhone podcasting app. You can look up This Week in Science.
That's right. For more information on anything you've heard here today, show notes will be available on our website. That's www.twis.org. That's TWIS.org. There you can also make comments and start conversations with the hosts and other listeners.
Or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twis.org. Just be sure to put TWIS, TWIS, somewhere in the subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for some more great science news.
And if you've learned anything from tonight's show, remember,
It's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just better understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
And that is the end of another show. This Week in Science, episode 485, is thus completed. What you got there, spiders? You show me spider pictures? I don't know where that spider went. I don't know where that spider went.
I have a collection of memes that I really like about this little jumping spider.
I love that. I've seen this before, and I love this little spider meme.
My family is cold and misunderstood. Can we crash in the corner for tonight? Or squish my wife. That's cool too. Hey man, there was a wasp in here, but I got him. I know how you hate wasps, and you know I'm always looking out for you. What's the book for? Hi, I just finished eating all the flies, keeping away the ants, and drove away all the termites. Oh sure, I'd love to read the paper. Hi, can I live in your spacious house? I promise to eat flies and keep to myself. Dear God, did you just spray me with furniture polish? You crazy?
I don't know what happened to that spider. It came down, it dropped from the sky.
He's watching you from afar.
I know, it dropped, and I saw it go behind the monitor, and I was trying to follow, and he went down in this little place between things, and then when I went to look after I finished the break, he wasn't there anymore.
Oh, speaking of this.
And that makes me really concerned, and I've been feeling like spiders are crawling all over me ever since the break.
Check this out. You'll
And
love
I swear
this
it was a brown recluse.
This morning, I'm me and my daughter are heading out to go to the zoo.
The zoo?
We go to the zoo 1 or 2 times a week.
He either said the zoo or they went to kazoo.
No, we went to the zoo. And I reach for my shoe and there's a spider web on it. But it's not the thin, wispy, regular, run-of-the-mill old daddy long leg.
Is it Black Widow spider web?
It's a thick, it's that thick.
That thick spider web.
Pencil strength to it. And I pull my shoe off and I feel the web and I kind of just take a little wisp. It's like connected to my shoe and I think, no, I know I'm overthinking this right now. There's no way that it's right now in my shoe.
It can't be.
So I turn the shoe and I whack it. And I whack it and I whack the shoe. And what do you think drops out?
A Black Widow.
Black Widow!
Oh my God.
And I'm thinking, oh, Black Widow, what do I do as I hold my shoe? Solution became rapidly apparent that there was a course of action that I would pursue in this.
Kill it with fire. The answer was kill it with fire.
Yes. I got a blowtorch. Went down to Home Depot. I kind of So I put it out of my misery.
A Black Widow.
But it was in my shoe. Like, I don't know if I'll ever wear that shoe. What if it made babies in there? Like, I'm afraid to go in my
There
shoe.
could be eggs in there.
I ended up wearing different shoes. Like, even though I'd gotten the spider out.
There could be something.
Throw them away.
Just throw them away.
It freaked me out.
Kill the shoes with fire.
And I love spiders. I don't, like we've discussed, I don't normally
not
harass
Black Widows.
spiders. Like, and actually, in any other situation, I would not have squished the spider, but because I had a 1.5-year-old like 2 feet away, I had no choice. I had to do it. I had to pull the
I would do it too. I usually let spiders go and don't worry about them too much, but it's when they're near my child's room.
Yeah.
In some way, I go after them. This spider, I didn't recognize it. I'm going to have to kill this house with fire.
So, I want to show you. I almost talked about the spider story this week, but I don't know. I'm not very well versed in spider neurons, so I didn't really think I could talk about it too much.
Oh, yeah, they recorded from spider visual neurons, right?
Yes, exactly.
That's so cool.
The method is really what got me crazy, but they were the little jumping spiders, which are peacock spiders are a type of jumping spider. So I have a soft spot for jumping spiders, obviously. Look at them. They're adorable. Look at that
They're
face.
so cute.
Oh my God. But so, here, I'm going to fast forward through these scientists talking so you can see the method, because it's pretty crazy how they did this.
Wait a minute. We have all this technology, and then he's showing a video on his phone?
Correct. So, okay, hold on. All right, so here we go. So here's how a jumping spider... So jumping spiders are special because they pretty much only use their eyes to find their prey.
Yeah.
Because they jump for their prey. Other spiders will use a web, and then they have the feeling in the web and all that kind of stuff. So watch this jumping spider jump. It's absolutely crazy. We've seen this video before, which I just love it. Yow. But now they're going to take the female jumping spider, and there she is. Whoop. And they put her in the fridge for a little while to cool her down, so she's slow. And then they dump her out. Then they put her on this little molded, 3D molded platform. Jumping spiders are bigger than I thought. They're pretty big. But so they have her stuck on there. She's cold, so she's slow moving. So it's like she's moving in quicksand. They stick her down, and then they stick her to the platform with wax. Poor thing. And so if you keep watching, you'll see she ends up with this hat of wax, kind of. See, look at it.
Aww.
So she's stuck. She's like, oh, what are you guys doing to
Really
me?
stuck there.
And then they poke a teeny, teeny, tiny hole in her head. So basically, when they've tried to read spider neurons before, they make a hole, and then they try to read in there, but because everything's pressurized inside a spider exoskeleton, pretty much they just bleed out immediately. Everything just kind of explodes out of the spider because of the pressure. And
That's right, because they don't have muscles. They have, it's kind of like hydraulic pressure.
Exactly.
Right.
So instead, they had to poke this teeny tiny hole. Look at him. He's just poking a teeny tiny little hole in there. And then they stick the teeny tiniest of electrodes into the hole. And then with this little girl waxed to this mold, they make her watch video of a fly. Then they can read what her eyeballs are doing. Look at that little hole.
That's tiny. Yeah, that's a teeny tiny hole.
Yeah.
That guy's like, I am good.
He looks like such a lab scientist too when he stands up in his cargo shorts. But look, see, here's the video. Is the little the fly, and then they have that little electrode in their brain, and they're looking. They're trying to see where she's looking. And that jumping spider is totally following the picture of the fly, and then they can measure which neurons. So it's kind of the start of it, but it's pretty cool. I love the methods. Look, you can see in their brain, whoa, looking at the fly. Oh, no fly. Nah, maybe, nah. Yeah, there's a fly. It's pretty crazy. So I love that, seeing the little jumping spider in the little contraption. There's something really cute about it. I don't know.
Yeah.
They just have the cutest little faces, jumping spiders. Even though the idea of a jumping spider is terrifying.
Well, yeah, the idea that there's a spider that's jumping at you.
Yes.
I like the idea of spiders that hang on webs, and that's where they stay. Not coming down at me from the ceiling. I want to see what kind of spider that was.
Although I absolutely hate when I have spiders coming down at me from above. That's terrifying to me. I hate it. I remember being a little girl and totally losing it, looking up and seeing a spider like here.
Right. Coming down. Stay somewhere away from me.
All right, so Blair.
Yeah.
I thought of a problem I have with the croc that chases the pig into the other lagoon with the other crocs. And it reminded me of this one time I watched some nature video where a pack of hyenas had cornered some sort of African buffalo, I don't know what it's called, and it was backed up in this little body of water, and really quickly, this crocodile from behind it grabs it and pulls it under the water. Huge croc. And the hyenas are still kind of waiting for this ox to come back out of the water. Some of them might have gotten a glimpse of it, sort of backed away, like, whoa, what just happened?
Yeah.
But they're all watching. Now, if you could just as easily say that the hyenas had coordinated with that croc to give it a meal, as I think you could say that the one croc coordinated to scare the pig into running into the water on the other side.
So let's see that 20 times, and then we can start to talk.
Right. So we got to see it 20 times.
So they saw it a lot of times.
Really? So
those are individual things. I mentioned 3 individual stories, but they saw in a statistically significant percentage of these videos and accounts that they got where there was collaboration.
Okay.
So it was this overarching theme in all of these accounts that they got back was that there was more than one crocodile, that they were working together, and in many cases that there was collaboration.
So the second one I got to show you these dolphins hunting on the shore, because it's actually a two-pronged thing. They're doing the corralling thing, then one comes across and sort of creates a starter wave, and then 3 more follow it to push that wave up the bank. So hang on, it's
So that's which one was that Kiki cited? Kiki, where did you find that thing where it listed all the different types of cooperation?
I put the link in the...
So this one's sort of interesting because it wasn't just like that they're working as a team towards a goal. This one's actually kind of synchronized. They have to be not just going for the same target.
Was one of the levels.
Yes, synchrony was one.
One comes across. Watch now. One comes across. It creates the initial wave. Right? He's kicked it with his tail. Right behind him, here come the other 3. And look how they're faced belly to back in order to create just the right wave, and so that they're not, each one can eat what's in front of them, otherwise, so they're not competing for the food. They've actually got all the same angle, and they had to get in there with the exact same timing.
Yeah.
And so it's a corralling, then a pilot wave, then this force up to the beach. This is just an incredible...
Yeah, so that's also incredible. I'm not saying it's not incredible, but it is.
I think you are. I think you're dismissing it.
Different from collaboration.
I think it looks collaborative. I think it's collaborative.
Each of them would be doing something different.
Then what was the other thing?
So there's coordination, where they concentrate in similar actions on the same prey and try to relate in time and space to each other's actions. They may begin from different directions or adjust their position to remain coordinated. And it's not incidental positioning to one another. If their approach, for example, if prey approach from different directions, individuals must have spread out at some point to positions that allow such an approach rather than the individuals arriving at the scene.
They have to stay in the same position. They have to stay in a formation.
Right, but it could still be coordination to do it, and then they share the prey at the end. Collaboration is that there is some sort of differentiation of roles resulting in team-like behavior.
And I think there is in this one. I would argue that this qualifies, but maybe not.
I don't think so. What is the URL? Did you put it in the chat room or something?
No, I put it in our the run sheet.
In the run sheet. Okay.
Next to your story.
I want this for myself.
Yeah, the paper looks interesting.
I closed it already. I have to open it again. It's like,
according to the study, millennials don't even know how to do laundry right.
So what is a millennial?
If you're born around the millennium.
If you're born around the millennium.
I'm guessing.
So millennials are in their teens still.
Let's see. Well, so this study did 18 to 33 years old. And that's, I guess, what they're calling the millennials.
Okay. I thought I was Generation X.
No, I was Generation X.
I'm Gen X.
Oh.
I'm Gen X. You are not Gen X.
Yeah, you're way too young to be Gen X. Don't get your own generation.
You're Gen Y.
Oh, I'm Generation Y, and then millennials are after me, right?
You're Generation Y me. Why me? Why do I have to inherit all of the problems that people created before I got here? Well, we all did.
Get out of here.
So I'm gonna just smoothly slide into cranky old manness as I age. I'm just gonna be so ready for it. You kids today with your loud music and your lack of morals and your iPhones embedded in your eye sockets. Why, when I was your age?
I think that spider was a brown recluse.
No, it's not. You're not in Texas, Florida, or Ohio, so it can't be.
Yeah, I don't think we get brown recluse in the city.
Hey, we don't get them in California. Actually, the city is the one place you might find them, though.
California.
It wasn't a daddy long legs, but it had longish legs.
No, it's not a California thing. It's
probably common house spider or something like that.
It's a Texas thing.
Brown recluse spider.
Oh, it's a grass spider. That's what it was. That's what it looked like.
Screen share.
They look like. If I can, yeah. Can I go to that image? No. Okay, so I'll scare this one up.
Brown spider.
So it was this spider that seemed to be coming from the ceiling. A grass spider. That's what it looked like. It had long legs like that, and I don't know how big those really are.
Gross.
It was not a wolf
It looks
spider.
like a wolf spider.
No, it was not a wolf
Okay,
spider.
I hate wolf spiders.
Potentially could have been this.
Did you just Google spiders in California?
Yeah, this is commonly encountered spiders in California, and it's a Berkeley website.
Cool.
So it looked kind of like it just had
Large
long legs like that, and then so it could also have been this Xeropsidae Mediterranean kind of spider, because that's another long-legged spider. It's so funny. Yeah, so this is what are supposedly commonly noticed spiders. So it's not a big deal, these spiders.
Brian Condren has given us a definition. If you became an adult since 2000,
That one.
you are a millennial.
Wah.
I guess I'm a millennial.
If you became an adult since the millennium, you're a millennial. Oh.
I don't want to be a millennial. They're a drag.
You guys want to keep looking at these
blame is all
spiders? I'm like, here, look at more spiders. Spiders in California.
I have to finish writing my script for this short we're filming because I want to send it to you tonight so you can look at it tomorrow and produce. I'm trying to figure out where to fit the interview in.
Aha.
I'm
Okay.
thinking in a few spots.
Okay.
It's
California
too long right
spiders.
now. I need to make it less long.
California spiders. I don't have to worry about it, right? I don't have to worry about it. It's probably that grass spider and not a brown recluse, even though
Brown recluse are usually not found in California.
I
They
know.
are usually in the south of California.
yeah, it's just the long legs, and I always look for the violin.
Kiki, on the other hand,
The violin is rarely found in California. A few cases have been found in the south of the state.
Yeah, they're not here. They're gonna get found. They're gonna fall off of cars that came from Texas.
Ugh.
You know, or you could have a neighbor who is really into spiders and wants exotic spider pets. Only sometimes they get away.
But they wouldn't do that with brown recluse. They're no fun.
Yeah, they would. If they're in San Francisco.
There's one in the San Francisco Zoo and the insect zoo. It's in a case inside a case.
We just got bats. We just got like 20 fruit bats.
That's cool. Oh,
And
the
an aardvark.
flying foxes. I love flying foxes.
And we got an aardvark.
You have an aardvark now at
We
this
got an
fox zoo?
aardvark. Don't get too excited. It's no more entertaining than having a kangaroo. They sleep. The aardvark just sleeps. He's super cute, and he sleeps actually right next to the glass where everybody can come see him. He like sleeps right next to that. He has a huge area, but he goes and sleeps right next to where everybody can come by and take a look at him. But he's so adorable and fat. He's a fat, round little guy.
I love aardvarks.
That's awesome.
They
Yeah,
look like hairless anteater pigs.
yeah, that's kind of what it is. Yeah, yeah.
I think they're adorable.
He is. It is a cute critter.
I got up close with an aardvark at the San Diego Zoo once. That was super fun. I poked his nose. I like to poke noses.
You poked an aardvark in the nose.
Boop. I like to poke animals in the nose. It's a gentle poke.
So birders have their life list of like birds they just want to see. You just have your list of animals you want to poke in the nose.
Yes. I've poked many of them already. I'm trying to think. Oh, copy. I still want to
Working
poke
at
an
a zoo,
owl
you get to
in the
do that.
nose. What else do I want to poke in the nose? So you've even poked some pretty dangerous animals in the nose. You just wait until they're tranquilized for a vet visit. Then you poke them in the nose.
And then you're like, poke! Gotcha! I got you in the nose! I poked a lion in the nose!
Yes, I did. I poked a polar bear in the nose.
I poked a shark in the nose! I know that Tony's Steele's not in the chat room, is he? He's
No.
not there. No. I threw up this picture during the end of the show.
Yeah. What is Slimer saying? I couldn't read it.
Slimer is saying, so it's a birthday cake. This is a birthday picture from Tony, and it says, Kai, should we be lighting your mom's cake like this?
What does Kai say?
And then Kai is saying, don't worry, who's the baby here? He's got his proton pack blaster.
Super cute.
He's going to light the cake. I love it.
Super stinking cute.
Yeah, and Kai saw it and was like, that's me, that's me. He was so excited. It was really awesome.
Are aardvarks the same as armadillos? No, not even close.
They're the same as millennials.
Yes. I could be an armadillo. I might be okay with it.
Oh, they have an armadillo too. We got to pet it.
Oh, do you guys... Oh,
there are 21 kinds of armadillos, you guys.
This one was tiny and had...
a lot of armadillos.
It was kind of scaly on the top and had cute little feet.
Okay, those are all armadillos. Did it roll up into a ball?
No, I don't think it was that big one. It was like a little
the ones
guy.
that roll up into balls are small.
Oh, it was still a pretty little guy.
And
but
there's only 2 types of armadillos that roll up into a ball.
we didn't see him roll into a ball.
So that it probably wasn't.
Did you
But they
guys... Oh, go ahead.
Yeah?
They were taking the zookeepers were taking it out for a walk, but not like on a leash. I guess they don't do that. They were just sort of like letting him run around on the grass, and then he'd go too far one way, and they'd sort of like walk over that way, and then he'd run back the other way. He was pretty quick, pretty friendly little guy.
Next year for Halloween, I'll be a millennial armadillo.
And we had what else did we get to? We got to meet a burrowing owl.
Oh, those are so cute.
Yeah.
I want to poke that in the nose.
I just shared it.
Remind me, Kiki, if I've ever got my children around Blair to keep.
Right,
keep her
she's
away.
gonna poke him in the nose.
I poked
She's gonna be like,
a
"Ooh, a
human
human
in the
child will
nose."
poke
No,
you in the
I
nose."
poked human children in the nose. That's not new. Turtle?
Porcupine?
I poked many a turtle. I poked porcupines in the nose. They're really fun to poke in the nose, especially prehensile-tailed porcupines. That is one of my favorite noses to poke.
Did you see the video that Matt Stafford sent to us?
No, I'm watching it now.
Yes.
I got it at work.
You have to. I'm not sharing it with everybody. I put the link in the chat room.
Do I have
The
a chat?
sound. The sound is what is really great about it, because this is a little talking porcupine. And he makes all these cute little noises while he's eating a little pumpkin. He's like, and the zookeeper is trying to say, "Can you say pumpkin?" He's like, it's got some sharp little teeth to be able to chaw away at a pumpkin the way that he does.
Porcupines are rodents, so they have ever-growing incisors.
Yeah, they're sharp. Could be sharp.
You ever see porcupine teeth, too? They're like orange.
Mm-hmm.
Covered in keratin instead of enamel.
Ah, that would explain the coloring. Thank you for that.
Yeah. That's a happy porcupine making this sound.
That's a talker.
He loves pumpkin.
That is not really what a porcupine sounds like, is it?
It is.
That's so
That is
stinking cute.
accurate.
Isn't it cute?
Oh, I miss working with porcupines. They're fun. They were one of my favorite types of animals to work with. Porcupines and alligators were a couple of my favorites.
And alligators. That's interesting. I understand the porcupine. I don't understand the alligator.
I don't know, something about the alligator. I think a baby alligator will pretty much always be my favorite education animal.
Hmm.
Yeah.
I can't hear that. That's awesome.
That's too crazy. Wow. Okay.
So cute. So cute.
Whiskey in the chat room has a question for you, Blair. Blair,
I'm
are
watching the
hedgehogs
porcupine.
and
I'm sorry. What?
porcupine closely related?
What? Say it
Are
again?
hedgehogs and porcupines closely related?
No, they are not. They are pretty much as far apart as two mammals can be. Porcupines are rodents, and hedgehogs are insectivores. So there's many differences. One being that porcupines, they can lose their quills. They don't shoot them, but they lose them. So the porcupine quills have basically little arrowheads on the sides. So when they poke into your skin, they absorb some of the fluid in your body. Those little wings pop out in your skin, and they're really hard to get out. And so they pull away in your hand, let's say, and they can let go of them just like we could pull out an individual hair out of our head. But hedgehogs can't do that. Those are just straight quills that are stuck in them. They can't release them in the same way. And they're insectivores, so they're related to other animals that eat predominantly insects. But the porcupine is related to guinea pigs, mice, capybara, all of the rodents. And they're characterized by their ever-growing front teeth and their herbivores.
Wow.
So that was just
knowledge download right there.
That was just the cursory explanation. I could get more into it, but I won't.
Why not?
No, it's all right. If you want to see a cool animal that's kind of like a hedgehog, but is a lot more unusual, it's called a tenrec, and they're closely related to hedgehogs. Yeah. Their little face is pretty cute. T-E-N-R-E-C.
How would you spell tenrec?
T-E-N-R-E-C.
That was pretty simple. All right.
So those are all close relatives to the hedgehog. If you Google image search tenrec, there's a lot of different kinds.
Those are all pretty adorable looking animals. I guess I have a. Is it just that they're mammals that I've got a soft spot for them? That can't be it alone. They're pretty cute.
No, I'm sure it has to do with their face. Dark eyes, big dark eyes. We did a study a few months ago. We did a study a few months ago about how animals that have facial features that are very similar to human children get similar responses, or that all baby animals have certain things in common to inspire parental care, and so there are some animals that maintain some of those characteristics later in life, and so they're considered, quote, cute.
Mm-hmm.
Right?
Right.
Are shoes insectivores? I think so. Let's look.
Shrews?
I think
Yes.
shoes are.
Shrews are insectivores.
Yeah.
Oh, no way.
What?
Well, I guess it's a baby tenrec. You know what it looks like?
What?
It looks like an aardvark. Maybe it
Let
is
me
an
see it.
aardvark.
Let me see it.
Hang on.
Screen share.
I'm bringing it. It might be, because this is... Oh, okay. It's pictures of cute and unusual zoo-born babies is where this came from, even though I was Googling the tenrec.
Zoo-born?
So I bet you it just is.
Zooborns.
I love
And-
zoo-borns. That's a good black hole I fall into often. I would recommend it strongly.
Okay,
Zoo-borns.
no, I probably shouldn't show you this. This is too much cuteness.
Show it to me. I can handle it.
I don't know.
Share.
Hang on. Let's see. How do I do this? How do I get it to do the right? Hang on. I think I gotta pull this out.
Tired, guys.
Yeah, I am tired. I know that for a fact.
So, Kiki, if I send you a script in like 20, 30 minutes. You'll check it tomorrow and
I will
respond.
not look at it tonight.
No, I don't expect you to look at it tonight, but tomorrow.
I will do my best to look at it tomorrow.
Okay, great.
Yes.
Oh yeah, that's a baby aardvark.
That's a
Jersey.
cute
Let me see
little.
that. Let me see that. Let me see that.
Oh my goodness, that is the cutest little guy.
It's so naked. Look at how
He's
naked
so
that little thing is.
Ooh,
He's so adorable.
ooh. Looks like a fetus.
I know. It's so wrinkly. It's like a hairless cat with a really weird face.
Oh, that was a tenrec.
That's
Yeah, here we go.
that's the tenrec. They're cute.
Those are being cute.
Baby. Wait.
Ooh,
Armadillo.
that looks like a
Aardvark.
deer, a mouse deer?
Deer mouse? It's part deer, part mouse.
No.
What the heck is going on there?
Not.
Are
It's
those
the
hooves?
smallest antelope. Yeah, it's hooves.
What?
The smallest antelope species.
No way.
Yeah, they're really
It
tiny.
looks. Wow.
If you Google search
So cute.
mouse deer, you'll see people holding them, and you'll see really how small they are. It's pretty ridiculous.
That's insane.
Ooh. Sloth.
Baby sloth. Oh, yawning baby sloth. Oh.
Yawning baby sloth made me yawn.
Oh.
Oh, wombat. I can't cuddle a wombat, but I can cuddle this plastic hippo.
What's that?
What is that?
What is that? That's not a.
I don't know what that is.
What do you mean you don't know what that is?
I think that's a cat.
That's a
No,
cat. Is that a
no,
cat?
it's a cat.
I think that is the worst cat ever.
That's the worst cat ever.
That's a baby
I
hippo.
think it's a cat. It must be a cat.
ever. Where's that? I have to find that blog again.
That
Worst
blog is
cat
one
ever?
of the
Is that what it's
funniest.
called?
Yeah, it's one of the funniest blogs ever.
Foxes maybe? I don't know what the yeah.
That's not what it's called. Yeah, those are fennec foxes. What's the worst cat? It wasn't worst cat ever. What was it called? Horrible cat?
Wow, what is going on here? How come the youngin?
That's a tapir. Yes,
Tapir. That's the one we talked about last week.
The
That
camo,
is
right?
related to rhinos.
Yep.
Yes.
But look at the broken up camouflage. I mean, it's got a ton of camo.
Distraction patterning.
Yeah, exactly. Disruptive coloration. We talked about that too.
Mm-hmm. Whoa,
Yeah.
that's pretty cool.
The worst cat.
Have you found it?
I found a blog talking about it. That's not helpful. Internet, you're broken. No.
I'll find it. I think it's on my. I posted it at some point last week. So. Right.
Tumblr thinks they may have found the world's worst cat. This is another article talking about it. I don't understand.
World's worst cat. I will find you for.
I know how you can find it. I've got it. I've got it dialed in now. I've got
You found it?
proper Google search.
I don't know if I think I don't know.
Just put in. Go under images. And then put in CAT. And hit enter.
Facebook, stop editing my freaking timeline. My timeline.
Technically, according to the legal department, it is not your timeline.
This is the best cat if you like your cats to be really sweaty and football shaped and disgusting.
You find it?
Yes.
Okay, good. Are you gonna screen share it?
I put it in the thing.
Oh, you just put it in the chat room.
Yeah. Oh my god, this is the best one. Alright, I'm screen sharing this. Here. And it says, "Cats versus dogs: the eternal battle." That little hippo and
That's
dog.
not a cat.
Where's cats? Oh.
I posted this picture before, and someone said this cat looked a little bit like steak. It does. It looks a little like steak. It's more rubbery than you'd like for a cat.
Cats just want to play and cuddle. This cat is dejected because it knows it is terrible.
Oh
Does
no!
anybody know what giraffes
Look, this is a
connection
terrible
between
hippo.
giraffes and our first walk on the moon are?
What?
It was a quote by one of the astronauts who was walking along, and he said, it's like we're in slow motion walking up here. It's like you ever see those giraffes in those wildlife movies when they've got it slowed down? That's what it's like. It's like we're walking like giraffes. Something along that line.
Huh? It's pretty weird. Purr. Now it's time for petting. This woman looks unhappy about her choice of cat. Honestly, I can't blame her. Oh my god.
I like that one. Ugh. Why?
Why?
And then the one.
Taylor Swift with a cat.
That one cracks me up.
I was saying this picture of Taylor Swift with her cat. I know she is a celebrity, but I really think she's setting a bad example by popularizing this breed. If you want a pet, there are lots of cats at your local shelter that are probably a lot softer, cuter, and also not as damp.
Oh, so cute. It's clear that what you in fact see here are cats. It's not very good cats. Oh my god.
Look at this one.
Can I have a hippo for Easter?
Yeah. The worst gift basket.
Please. I want a hippo for Easter.
This type of cat looks like it doesn't mind water, which is good, I guess, if you want to clean off the little bits of stuff that stick to it all the time. Oh.
I want a hippo. Can we have a hippo on Science Island?
Maybe a pygmy.
How much room do they need?
A lot. Yeah, let me in. I know some people are allergic to fur, but why does this cat have to be so sweaty? Oh, look at this one. Oh my god. Look at it. It's so cute.
All right,
Explode.
Kiki, Blair,
Yeah.
Indians.
Yeah.
It's 10:30. I gotta go.
Okay.
So do I. I need to go to bed. I am
Okay.
tired. My neck hurts.
Yeah, I'm eating dirt.
Yeah.
Okay.
I should have. I shouldn't have. I should have said it. Next Wednesday, we will be speaking with.
Mm.
We will have in an interview. Next Wednesday, we have an interview. Let me make sure I've got. That's what I'm getting to. Hold on. With Rob Manning, the Mars Science Lab chief engineer at NASA JPL. And we will be talking with him about his book, Mars Rover Curiosity. An inside account
Awesomeness.
from
Nice.
Curiosity's chief engineer. Yeah.
Do you have crib notes?
I will get you crib notes for the book. Yeah.
Great.
So we're going to be talking with him about that book. And then the 29th, we will be speaking with the lead for the Mars One survival. Their camp. What is it called? Let me make sure. Their simulation outpost project. So
Simulation Outpost Project. Now, exactly what is?
his name is Christian.
Okay, so on the 29th, I will be
What did you just
late
say?
or not be showing up because I am teaching a teacher workshop
The 29th.
on the 29th about how to teach children about climate change.
Right. I remember you said something about that, yeah.
So
Kristen von Bengston, so you won't be there.
I'm done at 7:30, but then I have to clean up, and then I have to drive home.
It's pushing it.
I might be able to join at like 8:30, 9 o'clock. So we'll see.
You need to clean up after the teachers?
Well, because I'm going to have like snacks and experiments and stuff.
Okay.
So I'm going to have to clean up, yeah.
Yeah. I haven't heard back from them regarding the interview on the 29th. They were asking because I guess Christian von Bengstrom is in Denmark. So he'll be a little off from our time. Kind of like Janiscu,
Yeah.
up all night. So they're asking if I can pre-record the interview with him.
Yeah.
Gotta be live.
We don't do that.
Yeah, let's simulate you're on Mars and this is the only time that you can talk.
Yeah.
Yeah, but anyway, fun Mars-related interviews the next couple of weeks. I didn't realize how much I themed it up. I didn't mean
That
to.
would be a fun
I
delay.
did.
Isn't that, I don't know, there's like a little bit of a delay there. I don't know if it's 2 minutes, 5 minutes, 8 minutes. There's a pretty decent delay, at least when they give a command to the rovers and it can enact that command. So
Yes.
that interview might be... I'm sorry, were you still answering the last question? You're still answering the last question.
Exactly.
And by the time you catch up, it would be just like the worst delay ever. It can be made to work with that.
You'd have to have scripted questions. You would ask a question, and then you would just wait.
Yeah.
You couldn't
But then
interrupt.
at that point, you could do one of those fake the two-camera interview, which I always think every two-camera interview is faked, where you have the interviewer standing here asking the question, but sort of in the foreground is like the back of the head and the ear of the person that they're talking to. I always assume that's a stand-in. They got like a body double who looks like the back of that guy's head, right? Or the back of that person's head. Like there's somebody sitting in a chair in the studio and they redo all the questions. So, what you're telling me is that blah, blah, blah with like the back of the head of somebody, it looks like in this example I'm giving you here, it looks like the back of the head of the giraffe you're talking to, but might not actually be.
Okay. Good night.
Yeah.
Yes.
I gotta go.
I'm looking at things. Ah.
But, there was something. Oh, it had nothing to do with me. That's why I didn't. Good night, everybody.
Good night, everybody. I've just had the screen on Blair because she's so pretty. Just everyone look at Blair.
Oh, Blair, one
everybody.
thing though, before you gotta,
One more
now that you've done that, you got a little thing. You got a little.
Right there. Yeah, right there. Just rub that out. There you are.
That's it.
All right.
Okay.
Did you have salad earlier? It was like that during the whole show. I didn't have time to point it out earlier, but.
Oh my goodness, you guys. Everybody thank you so much for the show. Thank you for joining us tonight. Thanks for watching. Have a wonderful night, everybody. Bye. We'll see you next week.